All right. We'll be looking at the eukaryotes cell division. All right. So we'll be very fast, and we need to make sure that we understand the topic. All right. So in it, we'll look at something like chromosome cell gets stages, mitosis. One of them will tackle. So this is part one. All right. Okay. So this is part one. You can see there. All right. So now, without even wasting our time, shall we start? Okay. So we are talking about eukaryote cell division, and we shall be looking at the division of the following. So suppose we have a cell like that. Let's say this is just an animal cell. All right. Inside there's what? A nucleus, right? So there's a nucleus and also just the cytoplasm around it, right? So a cell will divide like that. So that's a division, right? Then something that will divide, now we have what we call cytoplasm. So cytoplasm itself also will divide. So we have cytoplasm and also the what? Nucleus there. All right. All right. So the cytoplasm and the nucleus will divide. All right. So the division of cytoplasm, we call it cytokinesis. All right. So we call it cytokinesis. All right. Then the division of a nucleus, we call it, how do we call? Someone to tell me. All right. Not interkinesis. Oh, this one is called karyokinesis. So it's just karyo samplings like nucleus. All right. Then kinesis is just the division itself. Okay, so that is about the division that we'll be looking at. So we'll be looking at these types of division. So the cytoplasm and also the nucleus. So that's about the thing that we should be bear in mind as we'll be studying this. All right. So hope you have taken note of that. Okay. So now we are going to perform two type of division under eukaryotes. So Uh, one of them is what we call now mitosis. All right. So types of division, you have mitosis. All right, and the meiosis. All right. So these are the types of division that we'll be looking at. So in this video, it will be a very short video. So we'll talk about mitosis like that. Okay. So under this type of division, what's a division, by the way? A division just, or division of cells is just how cell divide, right? So we are interested, the cells should at least bring themselves on to life. That's the reason why we had one of the principle, or just the theory of cells, to say they come from pre-existing cells. How is that happen? It is because genes are being transferred from one generation to another. So in this study, we'll be there to study how offsprings occur in relation with a reference to what, to genes that are being transferred to offspring. So now that comes the issue of what, chromosomes. So chromosomes are the one carrying what, genes. All right. So these are just 3D like structure that carries what, genes, and the genes we are talking about here is what, the DNA. All right, so they carry DNA. They are just 3D like structure which was contain DNAs, and we are interested in the DNA which carries what, our genetic information. So DNA simply stand for deoxynucleic acid, deoxyribonucleic acid. So that's the abbreviation. Uh, that is it, the abbreviation for DNA. All right. So we said chromosomes will be carrying a lot of things, right? So we are talking about chromosomes. That's uh, that is something having our data. All right. So something that is having our data, meaning that we need to be interested in it. So we are studying chromosomes now. So we said these are just three like structure. All right. So they look like this. It be like when it's just one, you find it like that. All right. So once it is one, they at the mid, there is something that you're able to see. All right. That what we call what? A centromere. So that's a centromere. It is found at the what? Centromere. All right. So the other word for centromere, you can also call it kineteco. So this is where even the attachment of the other friends happen. All right. So kinetoco. So that's the other name for centromere. All right. Okay. So what if you find them now when they are two? These chromosomes when they are two. All right. So when they are two, the chromosomes when they are two, they will look like this. All right. So my drawing is just bent. All right. This is just bent. It's not supposed to be like this. No. So at this point, there is something that attaches them, and that's what we call the same centromere. All right. So that's a centromere. So then this that you can see on top there is a part of histone. So these guys, they are made up of a certain protein called histone, and also another part called what? DNA. So the DNA is also contained in it, and also so when you see these two now, this pair of seeing them on the opposite pole like that, we call them pair of what? Sister chromatids. So we'll say sister what? Chromatide. All right. So that's the whole idea about chromosomes. So these chromosomes that we say they are made up of protein. So they are made up of protein and also a DNA. So the chromosomes will be made up of DNA and protein. So um so made up of DNA and what? And the chromosome, like, and the chromosomes. No, and the protein. So they are made up of histone protein and also a DNA. So that's about the chromosomes. So histone is just a protein. All right. So now the other thing that these guys can do or can perform, the chromosome. So since it is made up of DNA and that chromosome, so in this chromosome we have about 40% of DNA in it. All right. So then we have what? We have 60% then of what? Uh, histone protein. So since now they have like this, so what you are going to do is that you find that this is a reaction, right? A reaction should have at least positive and what? Negative, so that they can pair. So this DNA guy will be charged negative. Then the histone protein will be charged what? Positive. Um, these ones are very simple. Histone is a positive recharge because of what? Arginine and lysine. So there is what we call lysine there and also ligin. All right. So arginine and lysine are found what? In the what? In the negative recalls of DNA. So um all right. So that is it. Sometimes it's not really that I'm sorry. So I've really made a mistake. I'm thinking something else. So the reason why they lysine and arginine, they cause the positive charge. So these two are what? Are for they are in charge of causing this positive charge you're able to see there. So then the DNA itself, it will be negatively charged. Why? Because of the presence of what? Phosphate. So there's phosphate ion or radical, which is what? Which is found in DNA. All right. So that is it. So the phosphate ion is going to cause the what? The negative charge in the DNA. All right. So um so that's how about to um that's about these chromosomes. So they occur in a reaction. So we can say we have DNA there. All right. DNA plus DNA plus histone, right? So DNA plus histone, they are going to form what we call nucleosome. So it's more like we are doing the condensation of chromosomes. Chromosomes start with the same this reaction, and we'll see how they'll be formed. All right. So we are reacting DNA, which is negatively charged, with the histone, which contain lysine and arginine, to form a nucleosome. All right. So what is going to be formed is what we call now a nucleosome. All right. So a nucleosome will be formed. So we can have a lot of nucleosome. Then after nucleosome, this once it grows again, it will go into what we call solenoid. Would have shown the structure of solenoid, but it's not really important to bring it to reality. We just have to know the reaction, because no one will ask you in an exam, draw the structure. All right, those are complicated things if we were to bring on board. So the other thing that will happen is that once the solenoid is what is formed, then it will after solenoid is formed, it will undergo again, it grows a bit, then forming something like a loop, and that's what we call chromatin loop. So it will form chromatin loop. All right. So then the chromatin loop now will result. It's a loop like that. Now this loop will result into what we call now chromosomes. So that's the reaction of chromosomes, guys. All right. So that's the reaction. So you need to know this. Someone will ask you um what's the product when or the hydrolysis or the growth of nucleosome result into what? It will result into solenoid. Solenoid result into what? Chromatin loop. Chromatin loop causes what? Or occur or just brings about what? Chromosomes. So those are the things you need to master when you are studying. Okay. So we are here to study everything. So that's how we have formed the chromosomes that you're able to see there. Okay. So we have talked about the structure now, how it is formed. Let's go now. We talk about the types. All right. Okay. Okay. Just a bit. Just a bit. All right. There was something, no? So now we have talked about this, and um we are now going to talk about the types of what? Types of chromosomes. All right. So we'll talk about the types of chromosome themselves. Okay. So we are talking about types or just classification. So these guys will be the same chromosomes we are from talking about. They will be classified as either um will have types, I mean names, but those names will be based on the positioning of the centromere. So the centromere, let's say we have these chromosomes, all right? Let's say again another chromosomes comes here like that. Then we are crossing these chromosomes. Let's say this, the centromere which was supposed to be here on the mid, it comes there, and this guy comes there, let's say. So we'll have something like this, all right? Then it attaches there, all right? Just a bit of it, of this one. This one is short, and this is the side we have. So it is short, right? We'll call that as a specific name once what it comes like that. Then again, when just the same centromere again matches on the middle there, so we have something like X. So on the middle there, we'll see the name how it will what to be caused as a result of the centromere be on the middle. All right, again it can be at the far end. So that's how chromosomes will be classified. All right. Okay. So chromosomes now be classified based on the positioning of the what of the centromere. So when we have the centromere at the center, we'll call that as the metacentric. So the metacentric is a situation whereby we have a chromosome there, all right? We have a chromosome there, all right? The centromere now will be just good on the middle, all right? So this is the centromere, okay? Okay. So that's the centromere. So that's a centromere. Then since that's a centromere, we call this as what? Um, we call this as the metacentric. It's on the on the middle. Okay. So that's what we call metacentric. Then when we have the chromosomes at the what? Or the centromere at the toward the end, is just toward the end like this. Let's say I have this. Okay, that's our chromosome we have. All right. So that's a chromosome we have. Then it's just toward the end. So what I'm going to do is the chromatin, let's say it's here. It was supposed to be here, but it is now here. So this is toward the end of this chromosome. All right. So we'll call this as what? Centromere. All right. So this centromere now, um, since it is toward the what? So at the center, this is at the center. All right. So you say when the sister or the centromere, or which is the kinetoco, is at the center. That's what we call now a metacentric. So at the center. All right. Then when it is just toward the end, it's toward about to go to the end, we call that as what? Acrocentric. All right. So that's what we call acrocentric. So when it is just toward the center. So we'll say toward the end, I mean. So when it is toward the end, we call that as the acrocentric. So what about when it's just um, 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 it is at the end, all right? So when it is at the end, so let's say we have this. So it has gone now, this centromere now, it is going here, all right? So for both chromosomes, it is at the end. So they are matching. Once it is this, it is reducing, and also this side is reducing. So, so, and also this is the centromere of this chromosome, that this end, right? It was supposed to be here when it was what? Metacentric. Now that it is here, and also for the other chromosome, it will be there as well. All right. So at the end. So this is the centromere, which we can also call it kinotoko. So get that. So uh, we'll call that as what? Um, we'll call that as the, we'll call that, once it is at the end like that, we call that as what? Telocentric. All right. So we call it telocentric. Okay, so it is telocentric. Then this is now at the end. Okay. So where that is when the centromere is at the end. So that's how we'll be classifying chromosomes like that. So once we classify them like that, it will even help us um to actually know the process called mitosis. All right. So that's about chromosomes. So we are done with the chromosomes. So now we'll go to what we call now mitosis. So we'll even actually know what a mitosis is and everything like that. So now that we have discussed the chromosomes and also the names, everything like that, based on the centromere, we'll go to what we call now um, mitosis. All right. So mitosis. So mitosis happen actually in the body cells or just the normal cells, and that's what we call autosome cells. All right. Autosomal cells are just body cells. All right. So these are just body cells. Autosomal cells, they are called what? Body cells. Okay. So since they are body cells, mitosis will be just occurring in those body cells, whatever body, apart from gametes. All right. So other than gametes, this mitosis will be taking place in those cells that you know. Okay. It will result in the formation of two identical cells. Let's say we have a parent cell. So this parent cell will separate like that. All right. Then once finally cuts and we'll have what? Two daughter cells. So these daughter cells, they will be identical. All right. They will be identical. So result in the what? Two daughter cells. So result in the formation of what? Two cells or just two daughter cells. So I'll be very fast now looking at the time. I'll be very fast. Okay. So the other thing that we can see here, in this type of division, yes, we said the two daughter cells will be formed. So these daughter cells, they will have equal what number of chromosomes? Let's say this cell had what? Had the four. 1, 2, 3, 4. All right, 4 chromosomes. All right. So once this one happens, it will actually, if they were four, these all these four will undergo the division, or it will just bisect like that and get what? Equal. So result in the number of equal chromosomes. So that's about two mitosis. It will result in the number of equal chromosomes. All right. Okay. So um all right. It will result in the formation of equal chromosomes or number of chromosomes. So the stages, it will undergo some stages. All right. So these chromosomes they were studying, they will undergo a certain stages or just stages, and those stages will present them or will abbreviate them as what? IS what? PMate. All right. So ISP mate, where I stand for interphase, this one is the synthesis stage, then P is what? Prophase, then M is what? Metaphase, then A is anaphase, then T is what? Telophase. So you remember how we were classifying um chromosomes. One of them was the metacentric, then acrocentric, then telocentric. You get? So those are the references that we'll be using now. Then we'll also know what is happening under interphase. All right. So stage one. So that's about two mitosis. So stage one, we call it what? Interphase. So what is happening under interphase? All right. So under interface, what will be happening is that we'll find some gates. So a cell, for it to actually grow like that, all right, let's say it is occurring in what is going through some gates. All right. So we'll call gate one, gate two, all right. So we'll call it, this one is G0, gate 0. Then this is gate what? 1. This is gate what? 2. Like that. So that's about it. That is under what? Mitosis stage. So we'll talk about the cell gates and also the checkpoints. So on as a separate topic. So the cell will be what? Will be going. There must be order also in the body for actually cells to be formed. There must be order. There are gates. All right. So the cell will undergo the gate, first gate, and that's what we call gate 0. All right. So it will just pass at gate 0 without anything. All right. So this is G0 or what? Gate 0, like that. Okay. So under this gate 0, the cell doesn't do anything. It is quiet. Sometimes we call this gate 0 as quiescent. All right. So you can be asked, what is the other name for what? What is the other name for gate 0? Is quiescent. All right. So that's the other name for gate 0. All right. So we are saying now no activities. All right. The cell is quiet on this stage, like it doesn't do perform, no change is happening, maybe just in short, no change is happening. All right. So it will undergo gate 0, no change. What about gate 1? So it will also undergo gate 1. All right. So that's gate 1. All right. Or um so it's called gate or gap. So they can be gap or gate. All right. So I'll say gap 1, gap 2. This is gap 0. So there are gaps here. So there's this gap 0, there's this gap 1, and also there's what? Gap. These are gaps, as you can see. There's G0 there, there's a gap, and also there's a gap there. All right. So those are the gaps I'm talking about. So I'm not really saying gaps as gates, but just know that as the gap. So gates will be checkpoints. So for now, we call them what? Gaps. It's a gap for the cell before it go into another what? Phase. And those phases, we call them the stage between these phases, we call them interkinases. All right. So you'll find even a question at the end. But they will have questions at the end. So we call it G1. So under G1, what is happening? Or just gap 1, what is happening? The cell grows in size. So cell grow what? Grow in size. Then the other thing that will be happening here is that um it will grow in size as to accommodate the organelles that are what going to be produced. So it need to grow so that other organelles which will come, it will be accommodated. All right. So that's about to the reason why it undergo G0, and we'll see why it is going gap 2. So gap 2 will be afterward the chromosomes is formed. So the second stage that I'll say is what now? Um, um, After going interphase, it will go what we call now synthesis phase. So I will name this as 2. So and that will be what? Synthesis phase. So under synthesis phase, the cell now it will actually undergo gap 0. All right. So this synthesis stage is what we call now the replication of chromosome. All right. So it is also called S phase, or just the replication of chromosomes will happen. So replication or duplication, but replication simply means a copy. So replication of what? Chromosomes. You see? So after that, once the chromosomes are replicated like that, what is going to happen that the cell now, after the chromosomes are produced now, the cell at least they need to check those chromosomes, all right? So that's where now they will undergo what? Gap 2. We remember we did gap 1 and gap 0. Okay. So under gap 2, all right? The cell will produce. So these are just gap, right? It's not a gate to say to be restricted, but it's a gap whereby it is taking changes. So under this gap 2, what will be happening that the cell reproduce organelles that will be led for what? Mitosis to really happen. So this is just a preparation of mitosis. So for the cell division. So the under gap 2, the cells reproduce organelles. We said here replication of chromosomes, right? So we say under this, we said the cell grows so that to accommodate this replication, you get. So now after this replication, what will happen is that the cell reproduce and um, the cell itself will prepare itself to actually undergo mitosis or the cell division process. So after chromosome replication chromosomes are formed, now we are ready, right? So we'll produce other what? Under gap 2, reproduce or produce. So the cell will produce other organelles or reproduce just organelles. So remember, we are done with the chromosomes. So we need something like cytoplasms. All right. So we need something like cytoplasms and other stuffs. So that's what we call produce organelles. After the chromosomes are there, and also the organelles are need to be produced. All right. Okay. So now from there, then what will happen is that the cell will undergo now what we call now prophase. All right. So the cell will undergo prophase. So under prophase, what will be happening that the chromosomes now will coil or they will at condense. All right. So chromosomes coil or just the formation or condense. All right. So once they condense like that, meaning that they become visible, all right. So the nuclear membrane which was holding them, it will also what? Disappear, all right. So the membrane which was holding the chromosomes, they are growing now, all right. So the membrane need to disappear. We don't need it because we're about to undergo what? Um, So it disappear or disintegrate. So we don't need the nucleus at this point because it will be holding the chromosomes inside from what replicating. So now it will disappear to allow chromosomes replication. All right. So we said chromosomes are the one in charge of our process of division. All right. So the spindle fibers, which is also called mitotic apparatus. So the spindle fibers, which is also called mitotic apparatus. So that those are just apparatus. Suppose we have the cell like that. So we have what we call spindle fibers here. All right. So at the end, those are the apparatus that we need. Then we also have so these spindle fibers. I'll talk about something else. Just wait a bit from there. So the spindle fibers what forms, all right. So they forms. So these spindle fibers, once they are formed like that, these spindle fibers we call them mitotic apparatus or gorgeous, but we can call them, they are just apparatus for what? Mitosis to occur. So they contain what we call microtubule. So microtubules will attach actually chromosome to the cytoplasm. So what will happen is that, let's say this is it is now at the center, right? Let's say these are chromosomes, all right. So they will be separated through these microtubule. So this is what I'm drawing are what you call microtubule. So this is the micro. So they are contained in these what? In these spindle fibers. So these are what you call spindle fibers. So pay attention to what I'm saying than to what I'm writing. All right. So that is it. Then the cell now so these is spindle fibers or microtubule. Microtubule will be used microtube. All right. So these microtubes will be used to actually attach the chromosome to the cytoplasm. So in this, these are the chromosomes, right? So in this is cytoplasm. So these spindle fibers will be used to attach from spindle fibers to what? To chromosomes. So that's what we call microtubule. So that's the charge of microtubule. They will act as if sometimes they act as filament, these guys, like to just to make the strength of the what? Of um the strength of um the strength of the cell. So that is it. So we go to a next thing. um we are now done with the prophase, right? So we now go to metaphase. So you remember, uh so this is now metaphase. So under metaphase, what happens is that the cell now, or the chromosomes will go at the center. That's the reason why we call it metaphase, metacentric from that word. You remember? So let's say suppose we have chromosomes like that. All right. Okay. So we have spindle fibers there. All right. Then the chromosomes are there, right? Okay. So the chromosomes now, this is now at the center. So we are now metacentric, heterocentric, acocentric. So they are now at the center, right? That's what we call now meta. So they are at the center. Okay. So what happens that at the center, when the chromosomes are at the center like that during the process of division or the growth of the cells, what will happen is that um, these condensed chromosomes that you saw, after they condensed, the nucleus was dis, it disappeared, or what it dis in integrated. So now what will happen is that these chromosomes now, they are what? At the metaverse stage. So they this at this point where it's straight like that, so we call it what? Metaphase what? Plate. It's a plate where they are found. So it's a plate, in fact, where they are found. So the chromosomes will align themselves at the metaphase plate. All right. So this is the metaphase plate. All right. So it's a metaphase plate. So metaphase what? Plate. So that's where the what? Chromosomes will align themselves. That's the meaning of metaphase. Chromosomes will align themselves at the metaphase what? Plate. It's a plate. Or they will go at the center. You can describe like that when you're explaining in your book. All right. So they will be caused at the center through the control of what? Microtubule. So microtubule will be what? Will be there to what? To bring these guys at the end, toward the end, at the center. So they're in charge through these pores called spindle fibers. So that's what you explain. So at the metaphase. So chromosomes, you say chromosomes uh, align themselves for what? At the center, or are pulled at the center. Okay, so once they are put at this, that's what you explain under metaphase. There's nothing much to explain. All right, you just say to them, they go at the metaphase plate, which is this plate you're able to see. They are pulled by what? By microtubules. all right, through the support of what? Spindle fibers. So that's about, we are done with metaphase. So let's go to anaphase, and that this was stage 4, right? This is stage what? 5, if I'm not mistaken. So we'll go to what we call now anaphase. So anaphase, so it's a phase um that actually we said ana acrocentric. You remember the way we were classifying um um the way we were classifying the centromere? You do you still remember? We said when it is just um at the center we call it metacentric, right? But when it is just toward the end, it's going toward the end, the centromere is going toward the, we call it acrocentric. Now this is anaphase relating to that. Okay, so what will happen that the sister chromatide, now that we saw, like the sister chromatids are pulled apart to the what? Opposite poles. So they were at the middle like that. So now what will happen is that at this point, stage 5, so we have the spindle fibers there. So these ones now, they will be what? Pulled. All right. So remember, this was what? Our metaphase plate. All right. So they are put like that at the end. So chromosomes are put at the end, or they are pulled to opposite poles, because this one we can see now is now, let's say, south. This is now what? Let's say north, or just east, something like that. Opposite poles, right? So chromosomes are what? So chromosomes, you are just describing the flow of chromosomes, guys. All right. So they are pulled what? In opposite poles. All right. So they are put in opposite poles like that. Okay, so that's what you describe under what? Anaphase. So there's nothing much, you are describing the flow of chromosomes. All right. So then the poles, now we have seen they have separated. This one is found, let's say, on south, and also this one on south, through the use of what? Microtubule, with the reference to what? Um, um, spindle fibers. All right, so that's what will happen. Then, apart from that, the pair of sister chromatide again separate. That's what we have seen there. They have separated the pair of sister chromatide. We said sister chromatide are these, right? So these are the one you are able to see. So this is what? Sister what? And this is what is here. Is what? Sister chromatide. So the pair of sister chromatids separate. That's the meaning. Or you can say chromosomes are what? Put in opposite poles. So pair of what? That's the meaning there. So are put to opposite poles, all right, or separate. So it's just the same thing. You are just describing the flow of what? These guys, the way they are leaving the center, which is the metaphase plate. They are leaving it and going to the opposite poles in the what? In the cytoplasm. So the cytoplasm at this point is also getting to separated, because as it's going this side, the cytoplasm, a bit of cytoplasm is pulled toward it as it is being pulled by the microtubule. So also this side, also there's a bit of cytoplasm is being what? Separated, or the process also what? Is being what? A carried of the process of cleavage. So it will be what? Cleaved like that. So that's about to the same stuffs. So it is at this anaphase where we even find a certain enzyme. So a certain enzyme called what? CDC what? 20. So it's called CDC20. This is just an enzyme that actually will cleave enzyme. So it will cleave, by cleave by I mean cutting. Okay, so I mean cutting the it will now cut the what? the proteins that what hold the sister chromatide. So let's say the sister chromatide are hold like this. This is the sister, right? These two are sisters. Okay. So they are sisters. So now what holds these sisters? Let's say these are sister chromatide. So there's a what? Something that to hold them there. All right. So it will actually, the enzyme called CDS20, it will actually what? Cleave the protein that is between here. In short, it's more like a groove. It was going there to cut that groove that hold what? The sister chromatide. So we are explaining this under what? Under anaphase, to show that they are leaving the metaphase. That's the reason why we are bringing all these points. Just to explain that we are leaving the metaphase with because through this supporting evidence, the enzymes, everything like that. All right. So meaning that this one also happen to start what? Separating the cytoplasm. All right. And remember, we talked about the separation of cytoplasm. So we'll talk about that at the end. So it's just trying to show now to say we have cleaved the protein that was holding the sister chromatide. All right. So we are done with that. So we saw the sister chromatide. Hope that is not giving you a challenge. The sister chromatide. So sister chromatide are just the, I repeat, this what you can see. This one, this point and this point, they are sisters, and also this point and this they are sisters. As you can see, they are going on the other side. They are going on the other side. So I want you to understand that next time when you know sister chromatide, don't get confused. So that's about sister chromatide. Then we'll undergo now the last stage, which is what? Telophase. So telocentric, you remember, whereby they go at the end and also finish the division. So those mitotes under this telophase stage, all right, let's say we have a stage like that. So these are what? Spindle fibers. Then microtubule, I'll draw them later. So what will happen is that the spindle fiber, since these guys have gone on the opposite poles, we don't need, they have helped us, the spindle fibers have helped us what? To pull them toward different poles so that separation can occur. So now what will happen is that these guys now, they are no longer in need, we are no longer in need of them. We don't want to perform end division after this, unless when other cells grows again. So what will happen is that the they would disintegrate or disassemble. So they would disassemble. So spindle fibers, this what? Disassemble. All right. So my handwriting is very bad, but you just get what I'm saying. So they will disassemble these. We no longer need them. They have helped us to pull things to separate a girl from a what? A boy. So we are done with this. So that's the reason why we no longer need them. So we also um form the nucleus, since these are sister chromatid. Now once they come like that as a coil like that, they will form again a what? A nucleus like that. They will have a membrane, and also the chromoside, the sister chromatid, which was it will form a what? A nucleus like that. You see the point? So these guys will be used to form what? A nucleus. Okay. So we can say the sister chromatid and also the sister chromatid, let's say they will coil like that and also coil like that, and they form a what? A nucleus, and this is the whole nucleus we're able to see there. All right. So they will come like this and also like that, and also that side they have separated like that through the what? The use of that. So now since that this side here, they will coil like that and also this coil like that, then they form a what? A nucleus. So that's what will happen. So the spindle fibers now, these guys are no longer in need, so they disassemble. All right. So once they disassemble like that, so the formation of a nucleus now that I've just demonstrated here. So formation of what? Of nucleus. So the nucleus now be formed. This is good. Okay. We are getting there. So in short, formation of nucleus, now we are saying nucleus reappear. Okay. That's the meaning. Okay, so once the nucleus is appear, or it is seen like that, the membrane also divide to form a cell. So the membrane, now this membrane, we're able to see this membrane there, all right, which is the membrane just. Okay, so it will divide to form a cell, so that it forms another thing. So this side will have a cell, and also this side will have a cell. So now the membrane divide to form what? So it is the cell membrane that we mean here. The membrane what? Divide as well. All right. It is now this final stage whereby the telophase, they are put what? At the end. So they are now going at the end. These guys, these chromosomes you saw, or sister chromatide, they will be put at the end, so that the separation what can occur properly. Okay, that's the meaning of telophase. So whenever you hear the word telophase, just know, go to telocentric like that. Okay, so what will happen that the 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 cytoplasm which was there also on the middle here, the cytoplasm which was there, it also what separate, and that's what we call, so let me demonstrate like that. So this is the final stage now. So we say the cell membrane what divide like that. So the cytoplasm which was there, it also what separate, and that's what we call cytokinesis. All right. So cytokinesis what happens at the telophase. So cytokinesis will happen then, um at this point when they were separating at this point, or just the nucleus when nucleus was dividing as well. All right. So when the nucleus was dividing as well at this point, because the nucleus now is dividing, so we'll also call it what? Or perform karyokinesis. All right. Which is the division of what? The division of nucleus. So we need to have two nucleus. So the division of nucleus. So that's the reason why we said nucleus reappears. So once it reappears, meaning it has divided. At first, it disappeared in the prophase stages. So now the karyokinesis is the process by which nucleus what forms. I mean divides. So that's the what? The meaning of karyokinesis. We saw this in the first page. All right. So this is about to um okay. All right. So that's about to mitosis. So in the next lecture we'll talk about so don't forget the 3DC20, which crave the protein that hold the sister chromatide. So don't forget that. Then this division we are from performing here, um it is called equatorial. So the reason why it is called equatorial, sometimes why is it called equatorial? All right, equatorial, equal things. So because it results into equal number of chromosomes in each two daughter cells. The two daughter cells, all right, they'll have the same number of what? Chromosomes. So that's the reason why we call it equatorial. Then for meiosis, just for your own information, is called reduction. All right. So this one is equal division. That one is a reduction, and we'll explain why it is a reduction division. All right. So it will result into two daughter cells. All right. So the ploid that we have like after the division, we'll be calling it as what? Diploid, because it's what? Two. It doesn't have the number of chromosomes. It bring the what? The same. So that's about it. Then we'll differentiate how we'll be using also the ploid of meiosis, which is just the haploid. But yeah, we'll talk about that. So make sure that you watch the video which is coming. So you can attempt these questions now. All right. So these are the question you can attempt. So describe the formation of chromosomes. So you write that reaction. Then the other thing that you can give, you describe the mitosis division. You write all the division. Then why is DNA negative recharge? So write some notes and send on my WhatsApp number. I'll give the WhatsApp number. So why is it on positive recharge? All right. Then the shape of DNA is called what? You need to find this one. How are the chromosomes formed? You talk about the reaction. What is the use of spindle fibers and microtubule? So you give these answers. So I'll need the answers on the line 09700200920. All right. So you can submit the answer through to this line. Thank you very much for watching the video. Please subscribe to our YouTube channel so that you can be updated whenever you post a new video. Enjoy the revision.